All right, back to this. All right, so hold on, not sure what the heck. All right, there we go. So the end goal of photosynthesis is to make glucose. And plants are super important to us, mostly because they generate oxygen. You know, everybody knows that. Additionally, they are the primary producers of glucose, which is hugely important for trophic food webs and energy transfer through food chains. Here is the reaction of photosynthesis. So it is a mixture of carbon dioxide and water and light create glucose and oxygen. This is a nice little diagram of that. You don't need to worry too much about the NAD pH part. So the total reaction is 6 carbon dioxide molecules, 6 water molecules, and light create a single glucose molecule and 6 oxygen molecules. And so glucose is created through photosynthesis, and then it gets used for respiration. So this is what the whole point of photosynthesis is for. It is to create energy. And that happens through the process of glycolysis, which breaks glucose down into 2 pyruvate molecules. And that reaction releases energy in the form of ATP, or adenosine triphosphate. You guys just need to know ATP. I'm not going to ask you for what that acronym means. This is the energy that cells use. Human cells, plant cells, any kind of cell relies on ATP and or ADP, but we're not going to talk about ADP, to utilize that energy. And glycolysis can occur with or without oxygen, and it takes place in the mitochondria. We're going to talk now about the 2 kinds of glycolysis. So the first is aerobic respiration. And this is when glycolysis occurs in the presence of oxygen. So in this kind of reaction, it converts glucose into water, carbon dioxide, and ATP. And that equation looks like this. So you have your initial glucose molecule that we had from our photosynthesis reaction, and it gets broken down into water, carbon dioxide, and 36 ATP molecules. So if you notice, that's like a pretty substantial number considering the amount of things we started with, which indicates that aerobic respiration is an incredibly efficient glycolysis reaction. And this is going to differ a little bit compared to our anaerobic respiration. So anaerobic respiration is simply glycolysis without oxygen. It is not to be confused with fermentation, although they are similar because they each produce ATP without oxygen. But there is some small differences that are beyond the scope of this class, but just keep in mind that these 2 are not the same, just similar. Fermentation can occur in both plants, animals, and microorganisms. And in microorganisms, a byproduct of that is ethanol. So that happens when we do things like brew beer or ferment wine. But it also happens in animal cells when we're producing energy. And we develop lactic acid. So anybody here who works out or is an athlete has probably heard of lactic acid before. That is fermentation occurring inside of your body. It can also be important for cooking. So really important difference between anaerobic and respiration. Anaerobic respiration and aerobic respiration is the amount of ATP produced. So anaerobic only produces 2 ATP per molecule of glucose. Remember, aerobic produces 36. So incredibly different efficiencies, but sometimes anaerobic respiration is simply necessary, either because these organisms just live in an environment with no oxygen, or in the case of people, when you're developing lactic acid, your body just has such a high energy requirement that you start undergoing aerobic and aerobic respiration to produce as much energy as possible. And that's why you get like huge buildups of lactic acid, because your body just can't process it fast enough. So those are the main differences between those 2. I believe that's it for today. All right, sourdough, we're not going to talk about this. This will come up next week, but this is going to involve some of that respiration we were just talking about. If you guys want to get onto some of these sites, I'll post the links on Canvas and you can start taking a look at it, but I'm not going to go over this yet. All right, let's talk about our homework again. First 5 INAT observations due by midnight. Please get those in. Your ecosphere setup and initial observations are due next Monday, February 15th. We have our next exam, or our first exam next Monday. And additionally, you have another set of INAT observations. So by midnight next Monday, I want to see a screenshot of 5 new observations, and there should be some indication that you have 10 total observations on your INAT page. If you just submit the same 5 pictures, you're not going to get points because we're going to try and get up to about 50 over the course of this semester. So that's that. Does anybody have any questions on the homework or on lecture? I'll post the slides again like usual. I know I just forgot to record most of that, so I will post probably the recording from Thursday's lab onto ours, just so you guys have a full recording. And that's my bad. If you guys notice I'm not recording, please feel free to put something in the chat or yell at me, because I don't always remember. What will the exam be based on? That was on the first slide I went over. It's going to be the first 3 lectures, so scientific method, biomolecules, and then photosynthesis, respiration stuff we talked about today, as well as potentially questions about your ecosphere and maybe INAT. So anything we've covered is game for the exam. All good. Again, the slides we posted, let's see. Please don't forget to be in class at 11. That's going to be super important for next week. It's the only time attendance is mandatory.
Oh, I have a question about the ecosphere.
Yeah, what's up?
Do you have like a size you'd recommend for the jar, like literage wise?
I used a gallon size jar, and I know that's kind of what a lot of people used.
All right, cool.
It's
Thanks.
pretty hefty. You can go maybe a little bit smaller than that. I think anything bigger than a liter is probably fine. Yeah.
All right, that sounds good.
Any other questions? All right, if that's it, you guys are free to go for the day. I know these are super short classes.
Thank you.
No problem, guys.
I'm sorry. I have a question about ordering the book
Yeah, what's up?
and the manual. I was just really confused from the module, and I tried to ask. Sorry, I lost my voice.
That's
I tried to ask some classmates via GroupMe, but no one answered. So for we order the manual through the link that you posted on the module, right?
I'm double checking Canvas just to make sure. Yeah. Yeah.
Okay,
right.
and then the algae, the beads, we order that too. But do we need to get the manual first? Where do we find the code?
The code is on the. Hold on one sec.
Because I think a few people are having a hard time finding it.
You get your manual. It is, of course, mine's out of order now. So it should be the 3rd page of your lab manual. There's a page that looks like this, and you get a code here.
Okay.
Everybody's code, I believe, is different, so you can't just borrow each other's code, because then if somebody plug theirs in, they're not going to be able to get their beads. So you got to use your own code.
So once I get the manual, I'll have the code for the beads?
Yeah, exactly.
Okay, awesome. All right, so I have to order the manual first, get that code, and then I'll order the beads.
Yeah, exactly.
Got it.
And
All
feel
right,
free
thank
to
you
tell any of your friends that who are asking.
Okay, I will. Thank you. I'll email you if I have any more questions. Have
Awesome.
a good one.
You
Thank
too.
you.
See you.